Good evening once again, everyone, and a very warm welcome to National Science Week SAEON webinar. I trust that you have been participating in the National Science Week programs and that throughout the week you've been making it possible through science. My name is Robotile Matawani and I am a SASTA volunteer based at the South African Environmental Observation Network. Our offices are in Gqeberha, and the node, as we all know it, is Elwandle node. So I'll be your host tonight, and I have no doubt that before we leave this Zoom call tonight, we would have learned a lot about how our day-to-day life as people affect our environment, and particularly the ocean and life in it. And I also know that we will learn more about how there's more to the ocean than meets the eye. I also very happy to get to learn that we have almost every members of our communities represented on this call, including our high school learners. So thank you so much for joining us. And please don't be shy to ask questions. In fact, I am asking you to please ask questions. Remember, there's no silly question. We're all here to learn from one another. But just before we begin, just some few house rules. If we could all please keep our mics mute at all times. If you have a question or you have a comment, you may use the reaction buttons at the bottom of your screen. You may raise your hand and I will ask you to unmute your mic and speak. But other than that, please use the chat box to write your comment, to ask your questions. But before we begin and our speakers getting ready to share with us what they know and us to interact with them, I would please ask the SAEON Elwandle node manager to please officially welcome us into this webinar. Professor Thomas Bonman.
Thank you, Madam Program Director. Good evening, ladies and gentlemen, colleagues, and distinguished guests, and a very warm welcome to the National Science Week webinar hosted by the SAEON Elwandle Coastal Node. This year's theme for National Science Week is Making It Possible Through Science, and the objective of tonight's webinar series is to illustrate the role of marine and coastal science in understanding the effects of anthropogenic or human-induced activities on marine biodiversity, with a special focus on microscopic plants, also known as phytoplankton, that forms the base of the marine food chain. So it's estimated that 30% of South Africans live within 60 kilometers of the coastline, and approximately 3.6 million South Africans depend largely on coastal food sources through subsistence activities. So our coast provides enormous benefits to the people of South Africa, and it offers many opportunities for future economic and social development initiatives. But our coasts are at risk due to increasing human pressure, and we will lose these benefits unless we maintain the diversity, the health, and the productivity of these coastal ecosystems. So the three talks in this webinar will give you some insights into the impacts that we as humans are having on the productivity of our coasts, and I trust that you will enjoy the evening with us. With that, over to you, Madam Program Director.
Thank you so much, Prof. I know now that we all know what to expect, and I hope that we are ready to hear more about how our lives affect the ocean and the life in it. But before we call upon our first speaker, there is a register in our chat box if you have just joined us. If you may please click on the link to sign our register, that will be very much appreciated. But for the sake of time, our very first speaker is an NMMU alumni. He was awarded his PhD in botany at the Nelson Mandela Metropolitan University, and this was back in 2014. He then continued with his research career as a postdoctoral fellow and a lecturer at the same university. His research Interest includes nutrient cycling, eutrophication, ecophysiology, bathing, and aquatic ecology, just to name a few. Right now, he's currently a biogeochemistry laboratory manager at SAEON in Note. But tonight, what he will be talking to us about is the causes and consequences of green tides in our South African estuaries environment. Thank you and so much, and over to you, Dr. Lucian Newman.
Thank you, Rebo. I just want to share my screen. Can you see the slideshow now, Rebo?
You're good to go.
Okay. Just apologies to everybody. My desktop at home doesn't have a camera, so I just put a picture of myself on so you can all see who I am. And as Rebo said, I'll be speaking tonight on the causes and consequences of green tides in South African estuaries. Rebo, you will give me an indication as to when my time is nearly up, I assume.
Yes, I will.
All right. Thank you. So just to get started, what are green tides? I'm sure everybody is quite familiar with the term red tide. Green tide, as opposed to red tides, are basically larger macroalgae that accumulate in the water column to such an extent that they form these mats, as you can see in this picture over here. And these blooms, macroalgal blooms, are often associated with eutrophication. So what eutrophication is, I'll delve into in the next slide. And basically, it's when water column becomes so enriched with nitrogen and phosphorus that it fertilizes, acts as a fertilizer for primary producers and makes them grow to extreme extents. Now, eutrophication can have detrimental consequences for water-related activities, in particular human activities. If you can imagine yourself in this estuary wanting to take a swim. So I don't think anybody would want to swim in an estuary that has a macroalgal bloom of such an extent. So it impacts on the aesthetics of the estuary and as well as the sense of place. But moreover, these things can have ecological impacts. So usually these mats occur in the spring and summer months, and then they die back in the winter months. This picture at the bottom here, this photograph, shows an underwater picture of how these mats basically sit on resident seagrass, which occur in estuaries. It's the prime producers of the estuary. They grow underneath the water. They are submerged macrophytes. And you can see this picture of this green epiphytized seagrass. So obviously that seagrass is suffering immensely there. It's not able to operate under its optimal conditions. So just to give you a bit of a background of what eutrophication is, now there is a definition, an old definition that I think is quite apt. It's the process of increasing the primary production of an ecosystem primarily through nutrient input. So where do these nutrient inputs come from? They come from various sources. Plants themselves or anything that decomposes and dies within a water column or anywhere else, even above the water, has nutrients within them. And when they decompose, they release these nutrients into the system in the form of inorganic nitrogen and phosphorus. But human activities can also contribute to nutrient enrichment in places like wastewater treatment plants, for example. They often release the effluent into coastal water bodies, and these are rich in nitrogen and phosphorus. So basically, these act as fertilizers for our water bodies. So what are the causes of eutrophication? It's increase in the input of inorganic nutrients to a water body, a decrease in the turbidity of the water, and also most importantly, a change in the hydraulic residence time of the water. What do we mean by hydraulic residence time? It's the time that the water sits in a specific area. Now in South Africa, there are different estuary types. You have open estuaries and you have estuaries which close off to the sea for long periods of time. Now, when the estuary is closed, that water body sits in one place for a very long time, and that means that it has an increased residence time. Okay, so very important to note, these two key parameters will be key in this talk. You've got increased nutrient supply, so more nutrients. If you can imagine, if you fertilize your lawn at home, for example, you treat it with fertilizer. Fertilizer basically contains nitrogen and phosphorus. So you're feeding the plants at home, your plants in your garden or the lawn at home. You fertilize it with nitrogen and phosphorus. You'll allow it to grow because you're giving it nutrients. So an oversupply of this leads to eutrophication. Okay, so often associated with eutrophication is hypoxia, or lack of oxygen. So the oxygen decreases in the water column. We call that hypoxia. When there's no oxygen in the water column, it's called anoxia. And this then can result in fish kills. So often you hear of in an estuary or in a river, there's been a fish kill. And probably the main reason behind this is that there was an oversupply of nutrients. The prime producers use up these nutrients quickly. They grow and multiply to such an extent that they now take up all the oxygen in the water column. And when there's anoxia, the fish have nowhere to go, or the fish are trapped in that area, and they die. They go belly up in the water column. And this is often associated with eutrophication. So there are different trophic states that occur. You have an oligotrophic state where there's low nutrients in the water column, a mesotrophic state where there's a medium supply of nutrients in the water column, a eutrophic state that the water body means there's an oversupply of nutrients in the water column, and then the hypereutrophic state is an excessively oversupply of nutrients within a water column, okay, or an estuary or a bay. So if you can show that the prime production of the bay or estuary is increasing, then the system is said to be undergoing eutrophication. But you got to go through all of these states in order for an estuary to become eutrophic or hypereutrophic. Okay, so now we know what eutrophication is. It's an increase in supply of nitrogen and phosphorus to a system. So the first case study I'll be talking about today is the Knysna Estuary. The Knysna Estuary is an estuarine bay in the Western Cape. It is the only natural estuarine bay in the country. It is ranked #1 in terms of conservation importance. So in our country, we have a ranking of estuaries, and out of all the estuaries in our country, Knysna is ranked as #1. And this is primarily because of its high biodiversity. It's got a lot of biodiversity in terms of fish, birds, plants, and invertebrates. And then also from a botanical perspective, it's ranked #3 overall in the country. So that means that Knysna is a very important estuary. So what we have in Knysna is that about 2013, 2014, there was a bloom of ulva that occurred. And ulva lactuca is a macroalgae. This is ulva, and there we have some pictures to show you how the ulva looks. It grew to such an extent that it covered the water column. And this was associated with excessive nutrient enrichment. Okay, so in this study, what we're going to look at now is the causes of the macroalgal bloom and some of the reasons why it's still persisting in the estuary today. So here's Knysna, just to show you how the estuary looks and where we'll be talking about. You've got the riverine reaches at the top, and then between the two bridges, that's the N2 going over there, and this is the old railway bridge. You have the middle lagoon, the lower bay, which open to the sea, and then right over here we've got Ashmead Channel, and around Ashmead Channel is where the bloom is localized. Just to zoom in there to Ashmead Channel. So our study sites where we collected samples was close to the channel, the main channel here, and then site 7 over here is close to the main channel as you go out, and all around Ashmead, sites 1, 2, 3, 4, 5, and 6 were in the Ashmead channel. Now I'd like to draw your attention to the wastewater treatment plant here. Out of the wastewater treatment plant, there's an inlet known as Bongani Inlet, and Bongani Inlet drains whatever waste comes out of the wastewater treatment plant. And remember, that's around about at site 3. So as scientists, we go out and we now have to go and collect samples and test the waters to see what's in there. So what we do is we collect nutrient samples to see what nutrients, nitrogen and phosphorus, what's the concentration of that in the water column, and we collect it right around from sites 1 to 7. We deploy what's called a benthic chamber to understand what is the contribution of nutrients coming out of the sediment. We measure things like redox potential in the mud, and redox potential is a good indication of how healthy you could say the mud is, and a bad, not a bad redox potential, but maybe a highly negative redox potential will tell us whether the sediment is anoxic. Anoxic sediments favor the release of nitrogen and phosphorus from the sediment. Okay, then we also measured the delta 15N isotope of nitrogen in the macrophytes and the algae material in order to trace the source. So what is the source of the nitrogen? The isotope can tell us, maybe it can distinguish between whether it's fertilizer, whether it's wastewater, or whether it's just natural atmospheric deposition of nitrogen coming into the system. Okay, so now I'm going to take you through some results. Don't be scared. I'll guide you all the way through the talk. Here we have in the first graph, we have ammonium here. Just get rid of some of these things. Here we've got ammonium, which is a nitrogen compound. And we see that at site 1 and site 6, the ammonium is very low because they're close to the channel. And then as we move along to sites 2 and 3, remember site 3 was close to the wastewater treatment plant. And this is, we have some peaks coming out in ammonium there. We see the same for our phosphorus, where we've got this peak going right around. We've got almost like a bell-shaped curve. So you've got increasing nutrients going from site 1 to site 3. And then dropping off to site 6. At the same time, we look at the oxygen. You find at site 3, there's a very low oxygen compared to the other sites that's close to the main channel, which is well oxygenated. Okay. So now if we look at another study that was done many, many years ago now, in 2003, the same trend was picked up by a guy known that did his PhD at NISNA in 2003, and he also found a similar trend to what we found. His station A5 is our station 3, and there's same type of trend. But what I want you to note is that the magnitude of his scale and my scale is quite different. Mine goes all the way up to 140, and his only goes up to 6. So the take-home story here is that, yes, the water column nutrients have increased over the years. So there's more nutrients coming into the system. Okay. Now this graph is basically the Bongani Inlet. What I did here was I went up to the upper reaches of the Bongani and the lower reaches of the Bongani, and this is where the channel meets the Bongani. So you'll see a trend here of nutrients high up in the Bongani being very high, 995 micromoles, then 578 micromoles, and comes all the way down to 146 micromoles, which is still very high. And the same for the phosphorus. So this definitely shows us that the Bongani Inlet, which is draining from the wastewater treatment plant, is very high in nitrogen and phosphorus. Now remember I told you about redox potential and sediment. So the sediment here we found was highly anoxic. Highly anoxic sediments, not good thing for an estuary because it releases a lot of nitrogen and phosphorus into the water column. So then we did some benthic chain deployments. Don't worry about the graphs once again. I'll take you through what you need to know. And here we show that we'll compare what we did with what the other study did. And there's basically an increase over time, showing that there's a release of ammonia and a release of phosphorus over time from the sediment. We had the same trend. There's an increase over time, an increase over time in phosphorus. But once again, I want to show you the magnitude of the scale. His goes up to 12 and mine is already at 90. So what take-home story here is the sediment nutrient flux has increased, meaning that the sediment is also releasing much more nutrients, nitrogen and phosphorus, in what we did compared to that time in 2003. So there's been an increase again. Now, the last thing that we did, we wanted to now trace the source and have conclusive proof or evidence of what's happening. And you'll see my scale here. The P is for Phragmites, or my, what do you call, not scale, what do you call it? My key. The T is for Typha, U is for Ulva, is the macroalgae that we're looking at. Z is for Zostra. This is the seagrass that occurs throughout the entire estuary. And what you'll notice here is that in the wastewater treatment plant, all the values are above 10. Outside in Bongani Inlet, it's above 10. And then the Ulva all around the Ashmead channel is around 10, except for Zostra here, which is 7.3, okay? So now 10 is basically our threshold. All signatures above 10 parts per thousand for the nitrogen isotope indicate that it's derived from human or animal waste. So it's really a key indicating that the wastewater treatment plant is one of the causes of the algae being around there, enriching the algae. So why do we have this difference? Well, Zostra is basically a rooted macrophyte, meaning that it's in the sediment, it stays there in the sediment, it can't really move, whereas Ulva, the macroalgae, is able to float around wherever it wants to with the incoming tide and the outgoing tide. Okay. Then we looked at what the impact on the salt marsh is. Now, the salt marsh is basically the natural vegetation that occurs in an estuary. An estuary is a highly saline environment, so not all types of plants can grow there. There's only plants that are specifically adapted to grow in saline conditions. And these are called salt marsh plants, or they occur in high salinity. So I'll only present now transect 1 and the control transect. The control transect basically shows you, there's Rebo in the picture. Can you see Rebo there? Back in the day, Rebo. So here you can see the control transect would represent our zonation of what should be going on in the picture. Or in the transect, you have a definite zonation of different plant species that's adapted to growing there. We've got Bassia, Limonium, Sarcocornia, Spartina, these, and then the seagrass, Zostra capensis. So that's natural. So the transect runs from the inside of the salt marsh to the water's edge. And here we've got another transect that's in the impacted site where the macroalgae is growing, going from the landward side into the water's edge. So going out from here to the water's edge. And as you can see in that picture there, the algae actually covered all of these salt marsh plants. So you can imagine how these plants were suffering under those conditions. Now, if you look at the green here, this Ulva lactuca, you can see it taking over most of the transect, okay? So under normal conditions, this Ulva would not be here and the plants would be growing happily. But because Ulva is there, it's smothering the plants and they are not able to grow to grow and multiply in the way that they should be. All right. So in conclusion, you have low oxygen in the water column. You have releasing sediments which are releasing nitrogen in the form of ammonia and phosphorus. And previously, Professor Allenson did a study there that showed Ashmead was shallowing, and it was these shallow mud-filled channels and that have this weak tidal flushing that are now able to support this anaerobic sediment, which then releases these nutrients into the water column. Okay, and I'll just leave you with a quote from what the studies in 2000 and 2003 said. They said that if either of these two features, the dissolved oxygen and tidal flushing, should change, the water column nutrients, ammonium and phosphates, would be expected to increase due to an increase in the flux of these nutrients from the sediments, which is what we saw in our study. Therefore, one could expect increased signs of eutrophication to occur, and this would be in the form of increased algal growth. And lo and behold, there we have a macroalgal bloom now sitting in Ashmead Channel. Okay, so the authors were spot on. Now, Rebo, how much time do I have left?
About 5 to 10 minutes.
Okay, so I'm going to stop my talk there. And I'm not going to give the other case study, which is very similar to this one in the Great Bruck estuary, which we also had a look at. And the main causes there were, again, nutrient enrichment from the sediments and a lack of flow. So a residence time, once again, and excessive nutrients contributing to a macroalgal bloom in the Great Bruck estuary. All right, thank you.
Thank you so very much, Dr. Newman, and thank you for that picture. And thank you again for letting us know what happened and why when we drive through the garden route, we see some algae blooming in the Nyasna estuary. Does anyone have any question for Dr. Newman? You can please write in the chat box, or you can raise your hand and then ask your question. Please do ask questions. No one's gonna think it's a silly question. It's just you wanting to understand and know, which is great. No question? As we wait for some of you to think about what they want to ask, if you have just joined us, if you could please sign the register in our chat box, that would be really, really great. And yes, please, you may unmute and ask your question, please.
Good day. Thank you. That was a very interesting talk. I was just wondering what possible solutions are there for the eutrophication. Yeah, I think one off the top of my head, I can assume that you should prevent nutrients to enter the estuaries. But yeah, I know at the Great Bruck estuaries there is a dam that they built that's now preventing flow. And yeah, I don't suppose you can just take down the dam, but what other solutions are there to the eutrophication?
Okay, thank you for that question. In Great Bruck, there's a study going on at the moment to reevaluate the estuary health. But in that system, we recommended previously harvesting the algae. It's not a solution to the problem because the algae will keep growing. But at least you can alleviate some of the nutrients, getting rid of some of the nutrients out of the system, because the problem there is actually the lack of flow coming in. As you rightly pointed out, it's the dam which changed the hydrodynamics of the system completely. When the dam was built, and still today, it basically took out 56% of the water that came through the system. So it's a very dramatic change. And unfortunately for that estuary, the only thing that will reset the estuary, if you will, is a big flood to take out all that organic matter that keeps refueling the algae. And our only recommendation at that time was to remove and perhaps harvest the algae. Because the algae is very high in nitrogen and phosphorus, it could be used as a composting agent, so maybe to fertilize as a secondary, what do you call it now? A secondary function, if you will, to maybe fertilize or use as a compost for gardening or anything else. But I'm not sure how. It's very difficult to stop eutrophication once it starts. There are some cases in the literature where like they limit the use of phosphate-based detergents. So that's been successful to some extent in international cases. But we need to, sooner or later, we need to start treating our wastewater, especially to a better standard, because even the standard that's allowed into the estuary, like of treated sewage, even though it's treated, it's still too high for our estuaries, because even the treated sewage, that the level they are treating it at, is still too high for our estuaries. So it will still be a form of nutrient enrichment, even if it's treated. But we need to definitely start treating our sewage better and limiting excessive nutrients into the coastal zone. And there's all sorts of activities that lead to high nutrients, not just wastewater. There's agricultural return flow. There's all these fertilizers, which can cause problems. So it's a huge social, economic process that we need to get to grips with. Any other questions, Rebo? Or did I answer your question?
Let's wait if she has a follow-up question first, or if no. Okay.
Hi, Lebo.
Hi. There are
Yes.
few hands that are. I have quite a few hands, and I also have few questions in the chat box. But I think we can just start with Gaddafi. If you could please unmute and ask your question.
Thank you very much. Am I audible?
Yes, you can go ahead.
Well, I was thinking, as naive as this is going to sound, but since the ocean is a larger body of water that is constantly moving, now can't there be a pipeline maybe that just directs the product or the water from the water treatment plant directly into the ocean, because the ocean can clean itself better than the estuary?
That's actually a good question, but the problem is the ocean also has. So this will be coastal. The water will be, the waste will be directed to the coastal ocean, which is just outside the estuary, and it also has its own unique flora and fauna, and the coastal zone as a whole is very linked. So it won't really solve the issue of enrichment, because what could happen out in the coast is that you probably, and the next speaker probably will talk about this, so I don't want to take his thunder away. You'll have some red tides that'll start to appear in the coastal ocean or coastal sea, if you will. But I don't want to go too much into that talk. The next speaker will highlight some of that. But in essence, it won't solve the problem.
All righty, thank you so much. Gaetano, I hope I pronounced your name correctly. If I didn't, please, I apologize. You can unmute and ask your question.
It's fine, thanks, Reba. I just wanted to find out over what period of time was the study conducted. That's question number one. Number two, what are the implications of climate change dynamics on the estuary and also on eutrophication levels? Thank you.
Okay, very good questions. Thank you. So firstly, that study was conducted in spanning from 2013 to 2017. And the second question, in terms of climate change is giving us, obviously there's a lot of unknowns with climate change, but for estuaries in general, or South Africa in general, I think in the coast, it's predicted that some areas will have higher increases of sort of stormy events and more droughts, and they'll occur at not a higher rate, but a more excessive rate, if you will. So more droughts means less water for estuaries, more stable water, like a longer residence time. More stable water bodies mean more a better environment for algae to bloom, or phytoplankton to bloom. And given the fact that there doesn't seem to be the pace of eutrophication doesn't seem to be slowing down, it's speeding up, it all leads to bad news for us, unfortunately.
Thank you, Lucian. Sabonga, you can please ask your question.
All right. Good evening, everyone. And yeah, thanks, Lebo, for the opportunity. Mine is very short. I just want to check from Dr. Newman if he hasn't considered to now compare the results across different seasons. And secondly, of these measured water parameters now, how far are they compared to the acceptable World Health Organization standards? Thank you.
All right, thank you. And so generally when we do our sampling approach, we take in seasonality into account, because we have to track when a bloom starts and when it dies off. So it's called a boom and bust cycle. So you got the boom growing phase and the bust dying off. So in general, we do sample over seasons to give us a clearer picture of the entire life cycle. And then your second question was regarding acceptable standards. So each country sort of has its own guidelines as to, they drop specific guidelines to adequately treat. In this case, I suppose you're referring to wastewater. Is that right? So I'll just go
Yes,
ahead.
that's right.
Okay, so so each country basically has a specific set of guidelines that are drawn up to treat their wastewater. Ours, even though it's the treated wastewater is an acceptable standard for how can I say now? It won't affect human health, if you will, but for our prime producers out there in our water bodies, the standard is actually still low. I mean, still too high to be meaningful. So it won't lead to low nutrients coming in. It will still be high. the standard is still too high for our prime producers. It needs to be even lower is what we would like for our estuaries and our rivers or wherever it goes into. But the standard is just a guideline. I don't think it's law yet. So there might still be ways to influence the guideline.
Okay, we have few questions in the chat box. And one of the questions to you, Dr. Newman, is that how can this increase in nutrients be reduced to have the seawater visible?
I'm sorry, Rebo, come again. How?
Oh, sorry. The question is, how can this increase in nutrients be reduced so that we can have the seawater visible?
I'm not sure I follow that question, Rebo. Maybe can the person that asked the question maybe just clarify so that I can follow it? How can the reduction in eutrophication or nutrients, what is it?
No, I think, I think it would be a good idea for the person to rephrase the question maybe in the chat box. And in the meantime, as we wait on the question, there are others in the chat box that I think we can answer. So there is a question that says, is there any conversation with various stakeholders regarding adjusting nutrient levels in the effluent from the WWTW? I think this is the wastewater treatment works.
Yes, so there are ongoing talks, and I think it's a process. Anything that we start with changing guidelines and so with different government departments is always a process, and there are talks that are ongoing. So, but everything takes time.
Yes, we have a hand. Please unmute and ask.
Evening, everyone. I'm Papama. I have a question for the speaker. So my question is just on a definition of estuary. Do we consider them as part of the marine environment or the marine body? So for example, what you're looking at, can you look at the similar thing in an ocean or a beach? So I want to know that when we're talking about the marine environment, can we include the estuaries? Thank you.
Yes, good question. Thank you. So an estuary is definitely considered part of the marine environment. It's the meeting place of the river and the sea. So you've got fresh water coming in at the top, and you've got seawater coming in from the mouth, and the two water bodies mix, and they form a very dynamic environment whereby you have different salinity. So fresh water has a salinity of 0, it's fresh, there's no salt in it. Seawater has a salinity of 35, so lots of salt in it. And then where the two mix is the estuary, and then you get this brackish water forming all the way from 5 parts per thousand or 5 salinity can be all the way to 35 salinity at the mouth. So there's a range of salinity, and this is characteristic of estuaries.
Okay, I think this one would be our last question, Dr. Newman. It's the question that we had previously. So just to rephrase, the question is, how can we decrease the nutrients in the seawater?
Nutrients in seawater.
Yes.
So, okay, so seawater I'm not sure. I'll just take it from what I understand from where the question is coming from, because it's a bit difficult. So in the sea, you basically have less of the land pressure as opposed to an estuary, which is surrounded by people and different activities. So in the sea, you have lower nutrients versus in the estuary that receives a lot of nutrients. I'm not sure if that was what the person was asking, but that's what I understand the question.
Okay, just to give you like a little information to go with when you are answering this question. So what the study showed is that there's an increase in nitrogen and phosphates over the years. And this caused eutrophication in the Naisna estuary. The question is then, how can this increase in these compounds be reduced? How do we make sure that?
Okay, so yeah, well, that's a good question. So the nutrients has been increasing over the years because of the activities around it. In Naisna specifically, a better catchment management strategy needs to be put into place. The water quality of the wastewater treatment works needs to be improved, and some novel thinking on flushing, which I'm not too good with because that needs engineers. Novel ideas on how to flush ash meat better. I would think some bioengineering would be good to come up with a strategy like that. But in terms of nutrients, definitely improving wastewater treatment quality, maybe upgrading the wastewater treatment works. I know the plant was upgraded before, but because the population of Naisna has now more than doubled to what it was when the study was conducted, it's probably due for another upgrade. All right.
All right. Thank you so very much for your time, Dr. Newman. I am sure that everyone learned a lot from your talk. Without any further ado, I will ask our next speaker to get ready. So our next speaker is an NRF Scarce Skills Postdoctoral Research Fellow at the Nelson Mandela University. He was awarded his PhD at the same university in 2018. His research field interests include coastal and estuarine ecology, eutrophication, microalgal dynamics, and harmful algal blooms, just to name a few. Today, what he will be talking to us about is harmful algal blooms in our estuarine and coastal waters, and maybe even explain to us why when we look at the water, sometimes the color isn't what we expect it to be. Over to you, and thank you so much, Dr. Daniel Lemley.
Thanks, Rebel. Firstly, can you see the presentation?
Yes.
I'll start my video as well for you.
Yeah, if you could just put it on slideshow, that would be great.
Which side are you seeing? Are you seeing the one with the notes?
Yes.
Yeah.
You put it on slide and not present view, I think.
Just click here. Which one is that?
Just click the one there.
That's the one that I did click.
Maybe click it again, then you go to options. I think it's under the display options.
This time? No, still not.
Just check display settings on top there, Daniel. Next to show taskbar and all that, end slideshow right at the top in on your screen.
Let me just try something quickly. Hold on.
As we're waiting for Dr. Lemley to get his slides together, If we could please have the register link in the chat box for those that came in late.
And now, Rebel?
Now you're good to go.
Good. Okay, so I think Lucian's probably given you quite a good background as to what's coming, but I think the difference of this talk is we're moving now from macroalgae and bigger plants that you can see with the naked eye. And now we're starting to talk about very small things that live in the water column, but are also plants. And that's the phytoplankton. So I'll be talking to you mostly about harmful algal blooms in South African estron and coastal waters. Just to sort of give you a refresher on what Lucian spoke to you about, one of the more recent definitions of eutrophication is that it's the enrichment of water by nutrients that causes an accelerated growth of algae and higher forms of plant life that produces an undesirable disturbance to the balance of organisms present in the water and to the quality of the water concerned. I know that's a mouthful, but ultimately it's nutrients that cause a primary producer response and ultimately an undesirable disturbance to the balance of an ecosystem. And just above there gives you some examples of what these consequences can be. Initially, you get a surge in phytoplankton or macroalgal blooms, as Lucian mentioned. One of the next knock-on effects, you can start getting harmful or toxic algal blooms, which leads to oxygen depletion, loss of submerged macrophytes, fish kills, and in very severe cases, you can even get the formation of dead zones. So because this whole talk and this whole webinar is about sort of the link between humans and the sort of the phytoplankton and the plants in this case, well, for all of the talks today, nutrient loading is key to all of this. And the identification and quantification of nutrient loads is key to the effective management of these systems. And this allows you to prioritize and also assess vulnerability in certain ecosystems around the coast. And that includes the estuaries as well as the coastal waters. So to start you off with, we will go first to Algoa Bay, which we are here at Nelson Mandela University, and we've got a lot of research going on. This was a study that I did in collaboration with SAEON, using all of their long-term monitoring data. And the reason that we started looking at the study, one, obviously we have a lot of resources here, but in 2014, we had a very big bloom and an unprecedented bloom along the entire coast of South Africa of this little dinoflagellate down here called Linguladinium polyhedra. And even though that this spread along the coast, I think it was from like Port Alfred all the way down to the Western Cape, this was one of the few places in Algoa Bay where the species actually came back a few years later, not to the same extent, but it did turn the waters another red color. So we were trying to understand possibly what could be the reasons behind this. Why is it specifically reappearing in Algoa Bay and not necessarily anywhere else around the coast? And so the way to do this was, as I mentioned, you need to know where your nutrient loads are coming from, who are the key contributors to this. And in this case, around Port Elizabeth or now Kabeja, wastewater treatment works or wastewater treatment plants are the primary source of dissolved nitrogen and phosphorus, which are your key sort of food sources for phytoplankton. The two estuaries, the big estuaries in the bay as well, which is the Swartkops and the Sundays estuary, were also highlighted as being very important contributors of phosphorus and nitrogen respectively. And in particular, when we're talking about the wastewater treatment works, there's two very big plants, the freshwater flats, is in a domestic and an industrial. And these are the key contributors to nutrients, and they go directly into the bay. They don't go via estuaries. So if we're trying to understand what, if anything, changed post-2013 when that first big bloom of Linguladinium occurred, in a nutshell, I know there's a lot going on here, but all you really need to focus on is from pre-2013, there's been a big increase in the amount of water and wastewater that's being discharged to our bay. And on top of that, we've also now got an increased availability of nitrogen and a reduction in phosphorus, which is showed over here. So for all three of the big wastewater treatment works that pump into Algoa Bay directly, there's been a significant increase in nitrogen, and there's actually been a significant decrease in phosphorus. And while that might seem good, coastal waters generally they've got very low nutrient availability, and there's ratios involved. And when you've got nutrient limiting environments, they can't always access all the nutrients. So if you shift that ratio towards something that makes the food, i.e. the nutrients, more accessible to phytoplankton, you actually then are promoting, even by dropping phosphorus and increasing nitrogen, you bring that ratio into favor. So what did we then see in the plankton responses? Firstly, within the bay itself, the western sector, which is where Port Elizabeth is and where Nelson Mandela University is, and where most of the industrial inputs and stuff come in, had the highest productivity post-2013. And that was shown by a significant increase in zooplankton biomass throughout the bay all year round. Even though there weren't necessarily significant differences in the background levels of phytoplankton, the number of extreme outlier events that occurred after 2013 increased significantly too, particularly around the western sector. And the reason and the drivers of this occurrence is most likely that the species came long ago via ballast waters. It might have actually just been in background concentrations for a long time, and then it just took the right conditions, and now we've got this overload of the species that's now sitting in the sediment waiting for the right conditions. And then from what this study showed was that increase in nitrogen, even though it might not be the cause, it's definitely a cause now that's making it reoccur. So I'm more so an estron specialist, so we're going to now talk about how can we learn from some estron examples that we've done over the years. And I'm going to start with some of the permanently or predominantly open systems that we've worked on. The first of which is the Sundays estuary. This is located in Algo Bay as well. It's got an extensive citrus cultivation industry in its catchment. There's an interbasin water transfer scheme that is in place to meet because as I'm sure a lot of people know, down here in the Eastern Cape, particularly around Nelson Mandela Bay, we're in a drought situation. Water isn't always freely available. So in order for this industry to survive, we've had this interbasin transfer scheme, which brings water from the Orange River or from the Gerib Dam. And what this has resulted in is a system that's extremely regulated and seasonal, which is highly unusual for an estuary, because an estuary is meant to be this dynamic environment where flow changes consistently and your responses are meant to sort of be intermittent. But what this results in is a system that's permanently eutrophic with harmful algal blooms that reside within the system. And we've had studies that we've looked at an annual basis all the way down to an hourly basis, trying to identify what's driving these species. So the first of these, which you'll probably see quite a few times in this presentation, is called heterosigma akashiwo. And it is a rhaphidophite species of phytoplankton. It is this little guy over here. It looks like basically a bunch of grapes, and all of these little green things that you see are its chloroplasts. And because it's got so many chloroplasts, that means that it's got extremely high photosynthetic efficiency and is able to take up nutrients very rapidly. And that's why it is a lot, well, because of those traits, it contributes to bloom events where it is almost the only species present, which is extremely unusual. And that is showed here where we did a study over a month period, and you can see that boom and bust phase that Lucian was talking about. So you get that extreme hyperaccumulation, and then the decay phase where everything drops off, they run out of nutrients, conditions just become unfavorable. And then there's an intermittent phase in between where things might sort of sit in background levels, and then when conditions are right again, you get another bloom phase followed by that decay phase. And the reason, you can see why those decay phase happens is because there's a depletion of nutrients by the bloom. So it's got no more food to live on. So that's basically the only reason the bloom stops. But then as soon as the bloom stops, obviously those nutrients keep coming in, the conditions become right again, and it can do it again. Another trait or characteristic of the species that allows it to be so effective at being a harmful algal bloom species is that it's able to migrate through the water column. You don't really need to take too much note of this. That's basically just to prove that I'm not talking nonsense. But basically what the species does is during the day, the water is, because we're in an estuary, and specifically the Sundays, you've got a freshwater layer that flows over the top and you have a more salty layer underneath. And that's called stratification. And what this species does is it uses that stratification layer and it can swim from the bottom where it stays at night because it doesn't need to be in the surface. And then it'll come up during the day where it's able to take up nutrients and obviously light, which it needs to photosynthesize. And then as a not that it does it on purpose, but when it goes down into the bottom waters at night, one, it helps it stay in the estuary and not get flushed out. And two, it's the opposite to what zooplankton do. Zooplankton tend to go up in the surface waters at night. So by them going down, they're actually avoiding being eaten or grazed upon. The other thing that this study showed is it was the first time in a South African estuary where hypoxia, which is the depletion of oxygen, it was the first time that we've actually shown hypoxia being driven by a single species in an estuary, which is basically something in itself that makes the species harmful. Just because a species is harmful doesn't mean it has to be toxic. Not all of them are toxic. They just need to have basically a bad influence on the community. So this is some work that colleagues from the zoology department have done, where we've now tried to look at the effects of these harmful algal blooms on the higher trophic levels. And basically what this showed is, on the left-hand side is a study that's done by Eugen Bornemann as part of his PhD, and he tracked the Mugelcephalus, which is a mullet species in the Sundays estuary. And basically the take-home finding was that the species avoids these bloom areas. So basically it's interfering with its natural pattern that it would usually take up if the harmful algal bloom wasn't present. And obviously this has knock-on effects because this species is quite important as a prey species for higher trophic levels. So when you start disrupting the dynamics of a certain species, you're actually having a knock-on effect on the entire food web. And then going even a further step down from that is Gilchristella larvae, which is also a fish species, but this is now the larval stage. And you can see here, this is when the harmful algal bloom was happening. And as soon as that bloom kicks in, you get a complete reduction in that Gilchristella larvae. And that actually creates a mismatch between the Gilchristella and its key prey or predator species, which actually are most abundant during those times. So all of these are those fine little scale details that show you the harmful effects of that phytoplankton can have. The Sundays estuary is also sort of the gift that keeps giving. There's lots of different harmful algal species present in the system. There's another one, a dinoflagellate, which is this little sort of mushroom-shaped species down here. It also forms exceptional blooms of over 500 micrograms per liter. It's got similar characteristics in that there's vertical migration through the water column. Mixotrophic basically means that it doesn't only use nutrients to grow, it can also use bacteria or organic forms of nutrients, which aren't characteristic of all species. However, and the other thing about this, the reason I say it's a winter opportunist, it's extremely good at growing during cold temperatures when most phytoplankton aren't very good at growing. However, in 2016, even though all the conditions were right for the species to bloom, it didn't bloom. And the reason for this was heterosigma was still present, even though it was in much lower numbers. The competitiveness of that heterosigma species was enough to suppress the growth of Heterocapsa. That also then leads to the next one, and this is sort of, as you can tell by the caption, this is a phytoplankton species that actually isn't phytoplankton at all. It's a ciliate. So what this species does, they're not the clearest pictures, but basically they look like little ticks with wings. And they steal the chloroplasts from cryptophytes, which are a type of phytoplankton as well. And once they've stolen those chloroplasts, they are then chloroplasts, they are then able to act as phytoplankton themselves. And again, they can take up vertical migration and are capable of taking up organic nitrogen. However, similar to the previous species, this species also doesn't like it when other big blooms of harmful algal species occur. So heterosigma stops its growth. Big blooms of dinoflagellates also stop its growth. So in the Sundays estuary, this species, which forms harmful algal blooms elsewhere in the world, isn't able to bloom simply because of one species, which is heterosigma. Lucian was also touching, or someone asked Lucian about climate change and what the effects of estuaries are. And the Gamtoos estuary, which is actually just outside Algo Bay, but about the same distance away from Port Elizabeth as what the Sundays is. This system closed for the first time. It's meant to be a permanently open system, and it closed for the first time in 2018. And this was due to ongoing drought conditions, which we still actually have. They're possibly worse at the moment. But the dam levels in the Coca Dam, which is situated on the Gamtoos estuary, the levels went below 7%. The irrigation volume, obviously, because the dams are empty, meant that the farmers can't use water to irrigate anymore. But what that meant was that there was a reduction in irrigation volume as well. And when you irrigate water in the catchment, that water seeps back through and becomes a nutrient and freshwater source to the estuary. So in the absence of both of these, the estuary closed for the first time in 50 years. So there's a lot going on here, but basically this just shows you on the bottom is the distance. So on the left-hand side is where the mouth would be, and the right-hand side is where the river would be. This side over here is basically what the estuary should look like. So you've got your salinity gradient with the fresh water over here and the marine water, and then it mixes, and you can see it forms that stratification that I was talking about. Similar things with oxygen, nothing too bad. It's generally well oxygenated, and the phytoplankton biomass isn't very high because the system isn't eutrophic typically. It's usually a mesotrophic state. However, when the mouth closed, you can see the entire system almost became homogeneous with a little bit of a freshwater signal at the top. And what this meant was that we lost that gradient, which then caused severe oxygen depletion in the bottom waters, in the middle and upper reaches, and it also caused quite big bloom events in the upper reaches as well. And just to show you some of these, so during the closed phase, it became eutrophic, and it was dominated by a harmful algal bloom species called Prorocentrum caudatum. It used to be called Prorocentrum minimum. Not that many of you would know that name or that it's important, but that species specifically is potentially toxic. However, once the water levels got higher, they breached the system, and then the system sort of reverted back to being in its normal open state. So it went from being eutrophic during the closed phase, and then went back to being oligotrophic / mesotrophic once they'd opened the mouth. And then the last of the permanently open systems, and the one that I think a lot of us are quite familiar with, we're doing a lot of work there at the moment, and that's the Swartkops estuary, which is an urban estuary that has an emerging harmful algal bloom issue. It's extremely important from a socioeconomic and ecological point of view. And there are 3 wastewater treatment works that discharge into the top of the estuary. There's domestic and industrial discharges from different canals. Because of all of this, there's an altered hydrology, so water flow within the system. And we've got a system that's been permanently eutrophic probably for the last few decades at least. However, in recent years, specifically since 2019, we've had these massive, so this water here, it shouldn't definitely not be that color. And the reason it's that color is because of heterosigma. That species has made its way into the system, and with ongoing drought conditions, we now have consistent harmful algal blooms. There's hypoxia, we have fish kills, there's been a loss of seagrass in the system, and to top it all off, in the river parts of the estuary, or the upper reaches of the estuary, we even have water hyacinth. During this big bloom, you'll see we actually had the first mass fish kill event in an open estuary, which is not usual, because usually when an estuary is open, there's place for the fish to move away and get out of the way of the bloom. But as you can see, this bloom was so thick that the fish didn't have anywhere to go, and there was a massive fish kill. Even the crabs started coming out from underneath and trying to get out of the water because of the low oxygen and the fact that this species makes a very thick mucus, which can be a problem as well, as it clogs the gills and makes breathing difficult. So as you can imagine, this caused a bit of a stir in the local media. And it's something that obviously is the goal of all of this research, to try and understand these blooms and how can we make it better. So getting into the last part of this, well, I'll say the second last part of it is talking now about small microtidal systems that close. And these are the dominant estuary type in our country, and they basically close off to the sea, and they don't always have a connection with the marine environment. And because of this, they have a high accumulation potential, which means that when they're closed, there's nothing going out. So whatever you put in sits there, and that obviously isn't a good thing if you're throwing lots of nutrients in. And they're naturally stressed, and they're vulnerable to disruptions. And because of this, they sort of warrant special management interventions, which I'll touch on in the next few examples, which are the Zandvlei and the Hartenbos estuaries, where both have harmful algal blooms, they have anoxia and mass fish mortality events. So the first of these was the Zandvlei estuary, which is in Cape Town. You can see here, this is sort of Muizenberg down at the mouth. And historically, this would have just been a temporary open-closed system without any of these developments. And actually, there's very old photos showing that the system almost used to dry up naturally. However, since the catchment's been hardened by all of these, that's a marina that's been built within the system. There's no wastewater treatment works flowing into the system, but because it runs basically through Cape Town, there's a lot of stormwater inputs coming into the system. And they started having issues with water quality problems as a result. So what they do, you can see here, this is how they open the mouth. And they do this according to a plan where it doesn't just happen, it's all a legal process. And there's certain water levels that the estuary has to get to and certain times of the year. And in this case, that's mainly winter because that's when the most rainfall comes in. And the idea is you're trying to open the estuary to increase connectivity and make sure that when you do it, it stays open for as long as possible. So in a nutshell, before they brought in this mouth management plan, there's also a weir just below the mouth, which sort of stops water from flowing. So this mouth management plan, together with them lowering that weir, has caused a big increase in salinity, which you can see here by this red arrow going up from 2012, where now the system is a lot more saline than it used to be only 10 years ago. And as a result of that, we get a reduction in phytoplankton biomass. So that sort of just shows you the relationship. As you increase your salinity, the phytoplankton biomass tends to decrease. And more importantly, there's less frequent big blooms, which can cause you problems. There's also a shift in the community. So your freshwater groups, such as your diatoms, your greens, and your glenophytes, they've decreased in the system because it's become more salty. However, just because you're opening the mouth doesn't mean you've solved the problem. There's still the nutrients coming in. So even though the blooms became less frequent, there were still blooms of species that actually like a bit of salinity, such as your dinoflagellates and your promnesiophytes, which I'm going to touch on now. So the species here that was the problem is called Promnesium parvum. It's a golden algae. In 2012, they had basically all of this going on in the system, dead fish, water that, as you can see why it's called golden algae, it turns the water like a golden color. And it lasted for over a month. And the reason for this is the species also has certain traits that are more competitive than others. So it will take up all the nutrients. It then begins to, obviously because there's no more nutrients left, the other species can't adapt. But what this species does is as it dies, it starts taking back up its own nutrients. And prevents any other species from growing. So the only way that they could stop this was by opening the mouth. And as you can see, in 2012, we had this very big bloom throughout the estuary, and there's been small reoccurrences subsequently, but every time those little blooms occurred, they would then try and open the mouth. And what's happened is we've never had a reoccurrence of that event. So in this case, mouth breaching showed to be quite effective. The last case study that I'm going to show you is the Hartenbosch estuary, and this is on the south coast, close to Mossel Bay. This is somewhere where I'm still actively working. As you can see, there's lots of bad estuaries make for nice pictures. So that was good for this presentation at least, but obviously not for the environment. This system has massive, this is not even a eutrophic system, this is a hyper-eutrophic system. There's an extreme excess of nutrients, and it's temporary closed. And it's got a wastewater treatment works that pumps into the top. So because it's closed, every time every day that the water that's coming from the wastewater treatment works comes into the estuary, it fills up, and about every 2 months they have to breach the mouth. But that isn't helping the issue in the long run. I'm just going to push play in the background while I'm talking. That's a video of them just after they've breached the mouth. Basically, you can see the cycling of salinity that happens. So every time the mouth is open, you get these like peaks in salinity, but as the mouth closes, it becomes fresh again until they breach it. And they do this very frequently. There's probably been about 13 breaching events in the last 2 years. And because the system's hypertrophic, you get extremely big blooms that mess with the oxygen levels. So when you've got oxygen levels above 20 milligrams per liter, that's excessively high, and actually also has bad effects on the higher trophic levels. But the problem with this is when those big blooms collapse, you then get, basically there's no oxygen left in the water, And then you end up with this, where there's just fish lying everywhere. And then their knee-jerk response always is to open the mouth and think things are going to get better. But what that shows, this was the second fish kill event that we had, I think last year in, I think it was August. The difference here though is when the mouth is closed and fresher, this tiny little species called Nanochloropsis is dominant most of the time. And you have your more freshwater species. But as soon as you open the mouth, it's not like the Zandvlei where the blooms go away. The only thing that happens in the Hartenbos is your bloom changes to something that likes more salty conditions and stratification, like heterosigma. So the way forward with all of this is, one, we have to reduce the amount of nutrients that are coming into the system or into all of our estuaries around the country. And some ways that this can be done is through artificial wetlands. We have a small artificial wetland here at Swartkops, not that effective because it's too small actually for the size of the canal. We also have another way that you could get around it is to reclaim treated effluents, not for consumptive uses, but there are purposes like livestock watering and irrigation or even industrial uses. It's not water, especially in a semi-arid country where we don't have enough water, this water can be reused. For the small temporary closed systems, obviously you want to optimize the mouth breaching protocols in order to facilitate effective flushing. There's no point opening the mouth and then it closes 2 days later. You've actually achieved nothing. If anything, you've caused more harm. And one way to do this is through continuous monitoring, because this will help you for early warning signs of harmful algal blooms and potential fish kills. And then the last of the slides is, so the sort of take-home message from all of this is that harmful algal blooms are becoming increasingly prominent in our estuaries and our coastal waters. These species have diverse ecophysiological traits which make them very effective at taking advantage of the situation, which also means that it's very difficult to get rid of them because not intentionally, but they're quite clever for considering that they're just phytoplankton. So we need adaptive management interventions like we've seen in the Zandvlei and Hartenbos. Although sometimes these management interventions, you do have to think them through sometimes, because if you're not dealing with the source of the problem, you're actually just putting a band-aid on an open wound and hoping it's going to do a trick. Because ultimately, if we let all of our systems start moving towards, say, what the Hartenbos is, we risk shifting them into a new stable state where we lose all sorts of ecosystem services and possibly won't be able to return. That's what this diagram shows. Once you reach that tipping point where the pressure is pushing on it, once you get down here, it's very difficult to roll that ball back up to the top of the hill. And another thing that's quite important is we need hydrodynamic variability. I know it's difficult because we're in a semi-arid country, but damming and those sort of things don't help the case, and neither does climate change, as was shown with Gamtus, where you've got climate change interacting with damming and you get quite severe effects. So ultimately we need a catchment to coast approach, it's a lot to say, to management. And it's not going to be easy, but there definitely are ways that can be done. So thank you.
Thank you so much, Daniel, for that very informative and thought-provoking. But yeah, that was very sad to see that we have species that are struggling to get oxygen in their own habitat. But I know that there's solutions somewhere and we will find it, hence talks like this. Anyone who has a question for Dr. Lemding? No, no one. Oh, Mr. Gadiano, please unmute and ask your question.
Okay, thank you very much. I just wanted to find out with regards to the issue of sedimentation. May you kindly explain that a bit? I also wanted to find out what is the role of rising sea level, particularly on the opening of natural estuaries, the opening and closing of natural estuaries, and what are the implications for for some of the estuaries, I think, that you have looked at? Thank you.
Sure. So when I mentioned sedimentation with regards to the mouth management plan, the reason the breachings need to be tailored. So typically what happens is you try and time your breaching so that, one, that there's freshwater flow coming in and enough of it. So in the case of the Great Bruck, which Lucian was going to talk about, what they do is they release water from their dam, and at that same time they open the mouth. And then on top of that, you also do it on a spring tide from the marine environment, because you want maximum exchange and you want things to be flushed out. If you don't do it like that, ultimately what you get is you've got no fresh water coming in, you breach it on an unfavorable tide. All that actually happens is you're putting more sea sand or sediment back into the mouth, and then that actually makes it more difficult in the future to open the mouth. So that's where we're sitting with the Great Bruck at the moment. They've been breaching with the drought because they have to breach. It's what they've been told to do. But because there's not enough water to release, the mouth actually only stayed open for 3 days or a week last year, and more sediment came in, and now it's going to be even more difficult to breach the estuary. With regards to the sea level rise, I don't know the exact predictions, but you'd imagine if the sea level rises, that should increase the chances of mouths being opened from the seaside anyway, because you do get sometimes where the sea opens the estuary on a sea storm, because storminess is also meant to increase. So if we have increased storminess in the sea, those sea surges that come in will open the mouth from the seaside. Whether or not it will be enough to flush the systems is another question. But I suppose on the flip side, if you've got rising sea level, that also means more sediment coming in, which might ultimately just mean that the mouth moves further back and gets even higher berms.
Thank you so much, Daniel. We have one question in the chat box, and the question is, are there studies being done simultaneously to monitor the impacts of fish populations or the larger food chain?
From the fish, obviously we all have, as I mentioned, we've got colleagues here at the Nelson Mandela University itself that we all do research. So fish research is done by a few different groups in certain estuaries, but in terms of monitoring, that's mostly down to governmental departments. I know the Department of Forestry, Fisheries and the Environment, they are responsible for that. And one of our colleagues, Dr. Stephen Lambert, who works for DFFE, basically his job is to go around the country and monitor the fish populations in a lot of these estuaries, not all of them obviously, but a lot of them. So that work is going on and obviously is available if needed too.
Thank you. And just a follow-up question on that very quickly. So they're asking if you are aware if there's any impact on behavior because of...
Yes, it definitely, this would be more anecdotal, I suppose, my response. We haven't got any direct links. Obviously, one of the threats is if there's a toxic species, and luckily this heterosigma isn't necessarily toxic, but you do get other species that can be toxic, and what that then does is that can bioaccumulate into the food web, and the fish or the shellfish that they eat can then make the bird sick, and there are instances of that around the world. In terms of the behavior itself on a smaller scale, not necessarily killing birds, but obviously when these fish in the Swartkops, for instance, when we had the big fish kill in the massive bloom, the fish basically are in a completely stressed state and they are just trying to get oxygen. So all of them are just sort of sitting at the surface trying to breathe basically. And then that completely changes the bird behavior, as you can imagine. So as these blooms are happening, you actually get a lot of birds at the estuary because they're just all picking off the fish that are trying to survive. So it definitely is influencing their behavior. But in terms of the bad knock-on effects, we don't have any clear evidence for South African systems, but I'm sure it does have some sort of effect or can have some sort of effect.
Well, thank you so very much for your time and for the talk, Dr. Daniel Lemley. For our very last speaker for the evening, she has started her academic career at NMU, where she completed her undergraduate, honors, and master's degree. Her master's research was on microhabitat and biotic structure of stromatolite formation. Right now, she's currently a PhD student at Rhodes University, and she is based at SAEON at Elwandle Node in Port Elizabeth, as now we know as Gqeberha. Her PhD focuses on identifying phytoplankton species in Algoa Bay, and she uses genomics and visual methods to do this. And tonight, what she will be speaking to us about is what she has been doing as her PhD study. Thank you so much for making time to have a chat with us. Let us welcome Rosalind Gibb.
Hi, thank you, Rogel. Appreciate that lovely introduction. I'm going to share my screen quickly. So I study phytoplankton in Algoa Bay and the Agulhas Current, but I'm just going to talk a little bit about Hypoxia in Algoa Bay. So my name is Rosalind Gibb, and I'm based here in Gqeberha at the L1 node, SAEON. So a little bit about what phytoplankton is. Phytoplankton are single-celled organisms that are found in water bodies, and they derive from marine ecosystems. They're base of the food chain. I'm talking about the coastal system here, so they're base of the food chain in the marine system. And things like zooplankton depend on them for sustenance, and that affects the rest of the food chain that depends on zooplankton for the food as well. So because they're base of the food chain, changes in the community have knock-on effects on the rest of the food chain, which means we can possibly use them as indicators for environmental change. So the idea is if we can map the change in the phytoplankton communities, then perhaps we can see what the larger effects of those changes are, because phytoplankton are very responsive to environmental conditions, as we've seen with Dr. Lemley and Dr. Human's talks. At the bottom here, I just got a few different types of phytoplankton. These are just some of them. So we've got some cyanobacteria, little blue-green algae, diatoms that have silica cell walls. The flagellates are a bit more armored, and they've got these nice flagella which help them to swim around. Green algae are stocked full of chloroplasts, and copperlithopores have these nice calcium carbonate plates around them. They're under threat of ocean acidification at the moment. Really won't go into that. So basically in my PhD, we want to know who's there, what are they doing, what happens when the community changes, and what causes these changes. So the idea is we can identify the phytoplankton and see what happens in the changing environment. Okay, my study site's Algoa Bay, so that's just here off Gqeberha. And we have these 8 sites that SAEON monitors on a monthly basis as part of their long-term ecological research. I only use some of the sites, but I thought I'd include them all for the talk. So we go out on the boat, we collect some seawater. So this thing is called a CTD. In the middle of it, it's got some probes that measure conductivity, temperature, depth, that's what CTD stands for, as well as some other environmental variables. Each one of those bottles, so these bottles over here, we open them up, and they are set with a computer to trigger at specific depths. And what this does is it helps us to capture water at a certain depth, so we can capture that phytoplankton community without mixing the water up too much. Okay, here we go, sorry. So what I do then is I go back to the lab and I filter the water so that we can extract the DNA from the phytoplankton. Now, phytoplankton are very, very small. As you can see, they just form a lucky film on the filter. So how do we extract the DNA from such small things? Well, we get different types of DNA methods. So normally we have barcoding, which is your normal, you have your individual, and you know that it's, species A or species B. You take the DNA out of it and you say, well, what does the DNA look like? Okay. Metabarcoding is a bit more complicated, where you take many different barcodes from organisms that you already know. So you know it's species A, B, and C, and you look at the the barcodes from those organisms. Whereas eDNAs, which I do, so I collect a sample, an unknown sample. So it's a sample of water. All it is water, I filter it. I don't know what's in there before I go into my barcoding. And then the idea is to say, well, here's the water, what's in it? So we sequence all the DNA that we can extract from the sample, from the filter. And then we use curation and analysis techniques that help us identify who that DNA belongs to. It's a little bit backwards to the traditional barcoding technique. Yeah, and there's just some pretty slides about the analysis process, and yeah. And then I also collect samples which I preserve for microscopy work. So I make some slides and I look at them under a microscope. So these are my settling chambers. So what I do is I put the sample of the phytoplankton in here with some water and dye, and then leave it overnight so that it all settles down to the bottom, and then I can look at it nicely. And it looks a little bit like this. So these big things are Noctiluca. It's a harmful algal bloom species, as we've heard about them already today. So these are some of the diatoms I found. So these micrographs are taken with a scanning electron microscope. I wasn't going to go into too much detail about who they are. So if you want to know which is which, then I can tell you a little bit about that afterwards. I think they're quite pretty. And here's some more micrographs of the diatoms that I found in my samples. And then I did a work shadow thing, I think it was last year, the year before, and we collected a live sample that we looked at under the microscope. We managed to see this group of, so some diatoms form little colonies, and then when they want to move, they move around by producing mucus, and they glide along each other in order to move. So it was really cool to capture this on a work shadow day, because then the students could actually have a nice look at this live. Sorry for the bad video quality, just taken with my phone. Okay, so we also get some silica flagellates, which weren't included in that first image with the different types. So these are also silica based, like the diatoms. And then we get some dinoflagellates, as you've heard already from Dr. Lemney, a little bit about what dinoflagellates are. So why is it important to study phytoplankton? Well, as I said before, the base of the food chain, and whatever happens in the phytoplankton community has a knock-on effect on the organisms above it in the food chain. So when you change from species such as diatoms that certain things like to eat to more harmful dinoflagellates, then that causes a big change in the community. And what this does is it has an effect on us, because we rely on fishing and a lot, and we rely on the ocean for a lot of other things as well. So these changes can be catastrophic. And then these phytoplankton respond to environmental drivers, such as nutrient input, which we've already heard about, which is largely driven by us, and then climate change affecting the temperature and chemistry of the ocean has a huge effect on these organisms. And then in some extreme cases, we get harmful algal blooms like this. So this is 2014. We had this harmful algal bloom.
Sorry, Rosalind, just a moment.
Yeah.
If you could please speak into the mic. You keep moving, so sometimes we hear you, sometimes it's like you moving away from the mic.
Oh, sorry.
Yeah, that is better now. That is great, actually.
Okay, cool.
Thanks.
Thanks, Rebel. So as I was saying is that, you know, these nutrient inputs and changes that we cause, as well as changes affected by climate change, alter the phytoplankton community. And sometimes we get these huge blooms. So this particular year, I think the wind was decreased and the temperature increased, and the species bloomed and caused the oceans to look orange. It's caused by Lingulodinium. Here's a nice aerial photograph of the extent of it. I think it was worse than this, but I can't find a photo that's worse than this. And then this bloom was all the way from, I think, East London to about Mossel Bay. It was quite extensive. And in very extreme cases, it can cause fish kills, and some species are toxic to us as well. So it's very important for us to understand how the environment and how we affect phytoplankton communities and how these changes occur, so that we can understand what effect that'll have on the rest of the food chain and how it will change ocean communities as a whole. So this is a photograph I took in 2018, in February, I think, of a bloom. So you can see a little bit, the water is a little bit brownish there in the disturbance by the boat. And then when I looked under the microscope, I saw it was Noctiluca and Lingulodinium, these two species in the photographs, causing this bloom. Yep, and that's it. I'll leave you with these pretty photos, these artwork. Sorry, I'll leave you with this artwork that was created by someone else. Does anyone have any questions?
Thank you so much, Rosalind. Anyone with a question, please raise your hand or type it in the chat box. As we're still waiting for the questions to come up, there is a link that will be sent in the chat box, and that is our evaluation for any question? No. Thank you so much, Rosalind, for this wonderful presentation. And I hope that very soon in the future, we will hear another presentation from you. So thank you so much for taking time out to come in to chat to us about the beautiful work that you do for the science community and also for the community as a whole. Thank you so much again. And for everyone else, today is not the last webinar that SAEON is hosting. There is another one happening tomorrow at 5 p.m. until 7 p.m. But this time it will be hosted by SAEON Ekagasini Nodes in Cape Town. Please join us as we're going to have a very interactive talk where we'll be speaking about fisheries and its relation to sustainable use of ocean resources. There will be a link in the chat box that will be sent that you can click to register so that you can attend the webinar tomorrow from 5 p.m. until 7 p.m. So please join us. It will be very informative and very interactive. And you will get to understand exactly what is happening in the fisheries industry. But there's a hand. Thomas, you want to say something?
Oh no, it's just an applause. Thank you, hand.
Oh, sorry. I thought you were raising your hand. I'm so sorry. Yeah, thank you so much. So if there isn't any question for any of our speakers, I would like to ask Professor Bonman to please say our vote of thanks for us so we can bid each other good night.
Thank you, Madam Program Director. I would like to thank the three speakers for their very interesting talks and the excellent questions raised by the audience. You guys were great. I hope everyone has learned something tonight on how our activities on land can negatively influence both our estuaries and our coast. I'm also pleased that through the interaction between the speakers and the audience, we managed to get a clearer picture on what could possibly be done to reverse these impacts, in line with the National Science Week theme of making it possible through science. It is also clear that we need a lot more of this good science and scientists for us to truly make a difference in the face of global environmental change. And I hope that the learners that joined us for the webinar tonight will be motivated to consider a career in science. Thank you very much for joining us this evening. Thank you very much to the Program Director, to SASTA for organizing this webinar and the webinar tomorrow. And please join the fisheries and its relation to sustainable use of ocean resources tomorrow evening. It will definitely be very interesting. Have a good evening further. Thank you.
Thank you so much, Tommy. Fundiswa, I do agree with you. I hope that everyone does listen and understand what we do affects the environment, but it all starts with us. I hope that everyone who was here tonight can go and at least teach one, so that together we can be able to be knowledgeable about this, that we can make good decisions. But you have been a very awesome audience. I am so grateful and thankful that I've got to do this with you. So thank you so much. But please, before you leave the call, again, there is an evaluation link in the chat box. If you could please fill it in for us. But after you have done that, please, please, please keep safe, adhere to COVID-19 regulations, and have a very beautiful evening. Good night.